class. In today's video, I will provide you a brief overview regarding how strand invasion takes place during the process of recombination. Let's see. Let's focus on the first figure. As you can see, there is a sequence. I have stated this as the first homolog. Suppose this is chromosome number, I am stating it arbitrarily. This is chromosome number 18, suppose. And I am stating this chromosome as a single chromatid. So this should be the first homolog. Coming to, let's say, the homologous chromosome for 18, let's say it is chromosome number 23. Let's assume it in this way. So this is our second homolog. So there must be sequence similarity regarding these two homologs. So I have stated it, both the homologs having the same sequence, which is GATC, TTG, CCC. I have taken this portion out of the entire chromosome just to make you guys understand it. So as you all know, the first thing that takes place is double-stranded break in one of the homologs. So on the first homolog, in this first homolog, there lies a double-stranded break where this sixth position, suppose this entire segment has undergone deletion due to breakage. So now the sequence lies with GATCT, there is a gap, then GGCC. So this homolog is under double-stranded break. Whereas the second homolog, the other homolog, that hasn't undergone any kind of breakage. So it is intact. So for the first homolog, let's say at the broken portion, we can assume that this should be our 3' end. I can state it hypothetically, not in real terms, but hypothetically, the broken segment, that means if GATCT is a segment and there lies a break, so we can assume that hypothetically that GATC, it starts with 5' and the segment before the broken segment is 3', hypothetically. Similarly, after breakage, this strand starts with GGCC. So GGCC can be stated as the 5'. This concept should be taken under consideration. So first thing, double-stranded break in one of the strands. As the double-stranded break has occurred, there lies a gap, and due to that gap, we can assume for a single homolog that this gap has generated a kind of polarity. That means 5' GATCT, 3'. Again, from the second part, 5' GGCC. Same thing goes for another homolog, CTAGGA, 3' to 5', and 3' again, as the polarity is from 3' to 5', so 3' CCGG. So this 3' or 5' or this 5' or 3', as you can see in the figure, they are all hypothetically considered. They are all hypothetically considered for the sake of understanding. Why? Because as you can see in figure 3, what is happening? There lies 3' overhangs. That means 3' overhang means for generating a 3' overhang within a double-stranded break within a single homolog, we need to cut portions. From the 5' end. Let's see over here. So as you can see from the previous figure, GATCT. Now, as we have assumed this segment as 5' to 3', and this segment as 3' to 5'. So from the 5 prime end, certain portions, certain bases have been obliterated out. Then only this blunt cut can be converted into 3 prime overhang staggered cut. Then only this blunt cut can be converted into 3 prime overhang staggered cut. As simple as that. Just for this purpose, we have to remove certain bases from the 5 prime ends. Just like this over here and just like this over here. So now we have our 3 prime overhangs. But the other homolog, it remains as it is. Now coming to figure 3. Our first strand invasion has taken place. Now this is a bit complicated. You must observe it closely. Now we have GATCT, and as certain bases from the 5 prime end is being removed to generate a 3 prime overhang on the opposite complementary base. So we have CC on the other hand. Similarly, for the 3 prime end, we have CTA just to generate a 5 prime gap. Now this segment which has an additional 3 prime overhang CC, this additional 3 prime overhang CC, this will try to introduce itself within the second homolog. Now, within the second homolog, the first homolog is trying to enter. This phenomenon is called the first strand invasion. In most of the books, it is written or mentioned in the form of a strip. But for proper understanding, I have introduced bases. Let's see GGCC. Now GGCC, after invasion, as both the homologs, they are similar with their sequences, cause they are homolog of each other. That means this 5 prime strand will show complementarity with this CC overhang. This 5 prime strand of the second homolog, this 5 prime strand of the second homolog will show complementarity with the CC. Now this is our first strand invasion. Coming to our second strand invasion. So our first strand invasion has occurred where GG is there and CC has been introduced. Now coming to our second strand invasion where the second homolog has now entered the second homolog, this one, the second homolog 3 prime to 5 prime strand has entered into the first homolog, showing complementarity at this region. So it has entered. So first thing, on first strand invasion, CC has introduced itself. CC is there. It shows complementarity with the second homolog 5 prime to 3 prime strand. Now second homolog 3 prime to 5 prime strand will now enter into the first homolog. As a result, due to this kind of invasion of the second homolog, the repair mechanism will be activated. Cause as you can see, there lies a gap over here. This region is completely open. Similarly, this segment is completely open. It doesn't have any bases. But we all know that we need to repair this double-stranded break. So our purpose is to repair the DNA. So that's why what will happen? Against this segment, against this portion of the DNA, a complementary base will be added using the repair enzymes. A complementary base will be added on the first homolog, using this segment as a template, using this segment of the second homolog as a template. This is how the bonding has occurred. A with T, C with G. And similarly, for C, as it has another template and it has already formed any kind of bond with GG, being a CC overhang, the rest of the portion will also be repaired. The rest of the portion will also be repaired. Where G is there, A is there, T is there. G is there, A is there, T is there. So this is how the repair process has taken place. This is how the repair process has taken place. So I am repeating myself. First, double-stranded break, one base obliterated, one base pair obliterated from one homolog. Now, as this portion has undergone break, rather blunt end or blunt cut has been generated, we need to form a staggered cut. And as per recombinant DNA technology, we all know staggered cut must occur with 3' overhangs. In order to generate 3' overhangs, we need to cut portions from the 5' region. That is happening. So from the 5' region, bases are removed for generating 3' overhangs. Now, what is happening? Strand invasion is taking place where the portion of the first homolog is trying to enter within the second homolog complementary base. As you can see, CC has now entered into the second homolog and it is forming bond with the GG. Now, this is our first invasion. On our second invasion, the second homolog 3' to 5' has now entered the first homolog. The second homolog 3' to 5' has now entered the first homolog. And as a result, as both the homologs has been introduced, as both the homologs have been interchanged, now the repair mechanism can start. How? Using these templates. Template over here, as complementary. As a result, repair enzymes will come and they will form or they will incorporate bases against these segments. As a result, the entire portion, the entire homolog double-stranded break, the entire homolog double-stranded break will be repaired. So this is the overall basic process regarding homologous recombination. If you wish to know more regarding the enzymes involved or even the non-homologous recombinations, stick to us. We will upload a YouTube video based upon it on our upcoming sessions. Till then, stay focused, build concepts and track progress. Me, Shank Banerjee, founder and educator at Vital Science Institute, signing off.